In 1990, the biological sciences embarked on their most ambitious quest to sequence the 3 billion chemical letters that define our species. This was the Human Genome Project, a decade-long effort to read every single letter in the human code. To manage a task of this scale, thousands of scientists across the globe adopted a highly methodical, page-by-page approach. They spent years meticulously mapping each segment of DNA before they even began the final sequencing. At a 1996 summit in Bermuda, the project's leaders established a groundbreaking rule. Every 24 hours, all new genomic data, like these raw strings of multicolored text, had to be uploaded to the public internet for anyone to see. This meant the blueprint of our biology was declared public property. By making the data open source, the project ensured that the findings would benefit global research rather than being locked behind a single entity's paywall. But in 1998, that open source model faced an existential threat. Scientist and entrepreneur Craig Venter launched a private company called Celera, entering the race with a $300 million budget and a much faster timeline. Celera utilized high-speed automation. By deploying newer hardware like the ABI Prism 377 DNA sequencer, Venter's team began reading the code at a pace that threatened to leave the government's 15-year project in the dust. Venter's goal wasn't just to finish first, it was to patent the results. Celera planned to claim ownership over human genes, forcing any researcher studying diseases like muscular dystrophy or cancer to pay corporate subscription fees. If Celera won, the Bermuda Principles would be effectively erased. Our shared genetic heritage would transform into privatized intellectual property. Francis Collins, the head of the public project, realized his methodical mapping wouldn't hold up against Celera's speed. Under intense pressure to save the public data, he made a radical pivot to adopt Venter's own controversial method. It was called shotgun sequencing. Imagine putting a massive volume like War and Peace through an industrial shredder. Instead of reading page by page, they sequenced millions of fragments simultaneously. A computer scans them, finds overlapping letters, and rebuilds the original text. By the spring of 2000, the public project was moving at lightning speed, but they hit a fatal bottleneck. They had generated a mountain of shredded DNA data, but no one had an algorithm that could actually put it back together. While the lab machines were successfully reading the code, the project was staring at failure. Without a functional assembly program, their data was just a pile of useless fragments. To de-escalate the public rivalry, the White House brokered a truce. Both sides agreed to declare a tie at a joint ceremony in June, giving the public team a hard 4-week deadline to produce a finished assembly. The project was in a state of panic. They had less than a month to build a massive computational engine from scratch, a task many experts believed would take years. The effort was saved by a 39-year-old graduate student named Jim Kent. Working from his garage, Kent volunteered to write the code that the federal centers lacked. Kent's sprint was grueling. He spent 12 hours a day at his keyboard, working on a fractured sleep cycle and icing his wrists between sessions to stave off carpal tunnel syndrome. Just days before the White House event, the code successfully stitched the fragments into a complete genome. Kent's work matched Celera's output and secured the public draft. That last-minute, one-man coding marathon is the reason the human sequence remains free and accessible today, rather than being controlled by a single corporate entity. On June 26, 2000, the first draft of the human genome was unveiled. By declaring the race a tie, the White House officially ensured that the rough draft of our biology remained open source. Because the data was free and accessible, it triggered a massive wave of innovation. Laboratories around the world were able to integrate this new genetic map into their work immediately. This fueled the rise of a multi-billion-dollar genomics industry. It led to the development of precision medicine and targeted cancer therapies that rely on the specific genetic profile of individual patients. The project's true value wasn't just in the letters themselves, but in the frictionless access that allowed thousands of scientists to innovate at the same time. The technology born from that 1990s arms race has since transformed criminal forensics. Massively parallel sequencing, the direct descendant of the project's automated readers, is now a standard tool for investigators. It allows police to extract DNA from highly degraded or microscopic samples,
providing the evidence needed to solve cold cases that have remained cold for decades. This rapid growth in genetic tech has even changed how we train the next generation of scientists. Molecular biology is moving into virtual reality, where students can manipulate three-dimensional digital structures to master lab workflows.